Welcome to MSBTalks, a podcast about science for everyone. Our guest today is Dr. Costas Lyssiotis, who is currently a professor within the University of Michigan Medical School. His lab currently studies the biochemical pathways and metabolic requirements that enable tumor survival and growth using mass spectrometry-based metabolomics. This episode, we talk about Dr. Lyssiotis' journey through his scientific background, research interests, and thoughts on the future of cancer biology. This is MSB Talks. We're glad to have you. Now for our conversation with Dr. Costas Lyssiotis.
So, thank you for coming here.
Yeah.
It's awesome to be in your office. Before we get started on more sciencey stuff, though, I just want to know a little bit more about you, how you got interested in cancer metabolism, and how you came to do research.
Oh man, I'll try to keep the story somewhat short. So I grew up in Michigan, that's why I was interested in hearing about your guys' backstory. I grew up in Oxford. I went to the University of Michigan. I was a chemistry major. I got involved in research, like you guys did early on. And actually, so when I came to Michigan, I thought I was going to be a doctor. It was actually in my first semester. I took organic chemistry, 210 with Brian Coppola. I don't know if you guys have taken Orgo.
Yeah.
And I fell in love with Orgo. I'm like, this is my language. And I was just so inspired by Coppola. He's probably the best professor I ever had. Aspired to be a thinker, teacher, leader like him still. Now he's my friend, right? So it's weird how things can change in 20 years. So anyhow, I was taking Orgo with Coppola, loved it, and I went to him and I said, I came to be pre-med, I'm pseudo-pre-med track, but I feel like this is my passion. How do I become a scientist? Never having any exposure to being what a scientist is, growing up in a relatively small town. And he said, you need to get involved in research, and so I did what any perfectionist would do. I looked up at all of the chemistry faculty and emailed every single one of them. Said, I want to join your lab and work on research. Your research is really cool. Obviously, I made them individual. This was This was before AI, right? So I had to actually think and read some websites. But before I even got done with that process, one guy emailed. I was sitting in the SLC, Science Learning Center. Still called that, right?
There.
Yeah. Are you a PLSG?
Sorry?
Peer-led study group leader?
No, I just do peer tutoring.
Okay. Do you work with Joe?
No, I don't know who Joe is.
No, Joe Salvatore.
Is he the director of the whole
Yes.
thing? Okay, yeah, I know that name.
Okay. Worked with Joe back then also, and Joe's daughter worked as an undergraduate in my lab. That's a tangent. Anyhow, let me see if I can get back on track. Sitting in the SLC, sending out emails, and in the process of doing so, one popped in. A guy named Gary Glick brought me to his office. He was upstairs, literally. He's like, why don't you come up? I went up, and I didn't have to do any more emailing through the chain because he signed me in. I worked in his lab. I actually worked on a drug that inhibits the mitochondria for cancer. So I wanted to do chemistry research. I ended up doing this more chemical biology related research. Strange that I came back to working on mitochondria and cancer 20 years later. That was definitely not the initial design, but so I did that with Gary for several years, and it was going well. I clearly had an aptitude for research, and I asked Gary. I said, "Well, pretty good at this. What do I do?" He said, "You get a PhD." So I applied to PhD programs. I went to California, worked in a chemistry lab, earned a PhD. There we were looking for new medicines that turned skin cells, which are readily abundant, can be easily isolated and expanded. We were looking for medicines that turned those into the precious cells that we lose in degenerative diseases that do not expand, like brain cells and heart cells. So we found some medicines that were able to do that in dishes, in a tissue culture dish. This is still a really active area of research, not something I'm involved with anymore, but it was cool, learned a ton. And then things continued to go well, so I'm like, I think I need to do whatever's next in this, just sort of inertia take me. I'm like, what's after a PhD? It's postdoc. And I felt like I'd had my share of stem cell biology, regenerative medicine. I wanted to take my talents to a different discipline, my ideas, learn something new. And around this time, what we're calling today is the field of cancer metabolism. This is a field that was really popular in like the 1940s, 1950s, if you guys want to hear about that history and why it shifted why cancer biologists shifted away from studying biochemistry and metabolism to studying other things. I'm happy to tell you about that. But anyhow, there was a resurgence of interest in this around 2000. How do the metabolic pathways that regulate a cell's ability to grow and survive and resist treatment, how do those influence cancer outcomes, and can we target them? So I thought this is right up my alley. It's biochemistry, it's cancer biology, so this is sort of a new biological patient-related discipline, but it uses techniques and concepts in chemistry, which is where I was good. So I joined a guy's lab at Harvard who was an expert in this area, one of the emerging leaders. And well, I was in his lab for a couple years, and it was going well. Just a quick tangent. As I was in his lab, when I joined his lab, I said, what are the odds this guy's going to move? He says 0. I asked members of the lab, 0. Well, they were all lying to me, or things change. So I started my postdoc in Boston. It was also when we had our first kid, and then moved to New York City. So he moved the lab. He was the cancer center director at Cornell. Cornell Medicine is in New York City on the Upper East Side. Cornell's in Ithaca. So I finished my postdoc there with Lou, and at the end of that, it was the writing was on the wall. I was good at research. I had an aptitude. I enjoyed doing it. I thought I wanted to lead my own research group. So you go on the job market and look for places. And I didn't think I was going to end up back in Michigan. Like I told you guys, I was from Michigan. Spent some time in San Diego. Spent some time on the East Coast. I thought I was going to be either a warm weather guy or a city guy. But for a number of reasons we can talk about it, we ended up back here. That was 2015, and so I've been running a research group. You want the whole backstory, right?
Yeah.
I've been running a research group here for 10 years. The lab started working on metabolism in pancreatic cancer. So what do pancreatic cancer cells eat, and how does that shape their ability to make more pancreatic cancer cells, to evade therapies, to evade the immune system? Still think about those concepts, but now we also apply them to not just the cancer cell itself, but the cancer ecosystem that is also known as the tumor microenvironment. So cancer cells don't live in isolation. They have all kinds of friends around. Immune cells, stromal cells, vascular cells, neurons, fat, and all of these different cells that interact with the cancer cells influence how the tumor is a tumor. They also influence the metabolism, the local metabolism. The cancer metabolism influences the host metabolism. So physiologically, how does the cancer instruct the host? How does the host instruct the cancer? So those are the concepts we work on. We do this, like I said, in a major area of study is pancreatic cancer. I am the co-director of the Pancreatic Cancer Center here. We have a super awesome team around us. My lab works in metabolism, but colleagues in labs sprinkled throughout this floor and this building who work on other aspects of pancreatic tumor biology, and we're a great team. Like I said, I've been running the lab for a decade. The wet lab is here in the Cancer Center on either side of my office, actually, right on this floor. I also have a mass spec lab out on North Campus. It's where we do our mass spec-based metabolomics. So I have really talented technical staff that work out there with all of our really expensive, fancy instruments. Those instruments allow you to do what we call metabolomics. And so, if you've not heard of this term before, it's like all of the other omics. So if you talk about genomics, that's what are all the bases in someone's genome. ATCG, ATC. If you're studying the proteome with proteomics, what are all the different proteins in a cell? So metabolomics is the same, except it's a way to look at all the metabolites in a cell. I'm also director for the graduate program in cancer biology, so consider part of my mission to interact with graduate students and to train graduate students and lead the next generation of scientists. I think that covers the major bases.
It's really cool.
That was great, yeah. What's like an exciting avenue in metabolic biology that you're looking into right now?
Gosh, how do I pick?
Anything.
This idea that cancer isn't just so. When people think of cancer, when lay people think of cancer, maybe Anna doesn't because she works with pediatric, but they often think cancer is just this lump of bad guy cells. And that's true in part, but in the disease we work in, while there are those bad guy cells, they can constitute as low as 10% of the total tumor. So 90% of a tumor is not actually cancer. But if you act on that other 90%, you can influence the way that the cancer cells respond to therapy, grow. They can grow better, they can grow worse. So understanding those interactions is super important. And what we've learned in my group is that metabolites shared from all of those other cells can support the growth of the cancer cells, who can sometimes be limited in their nutrient availability because they're in this sort of atypical area where they don't have ready access to the bloodstream, and so the other guys around the cancer cells will share nutrients with them so that they can do the things that cancer cells need to do, primarily grow. If cancer cells weren't growing, we would never know that they existed. We would never care about them. The other thing that the tumor microenvironment does is it helps the tumor to pretend like it is part of the normal body. So our immune system is constantly surveilling self and looking around for foreigners, right? Virally infected cells, bacteria, and it's the job of the immune system to track those down and kill them. The same is true of tumors. So one of the tricks that tumor has to do is to convince the host that it's self, or to keep the ability of the host from killing it. All right, so that's really multifaceted, and they do ultimately come out to be one and the same thing, though there's certainly some nuance there. One of the ways that the tumor is able to trick the host from being able to kill it, so the immune system has killer cells in it. Big player in that are called CD8 T cells, or cytotoxic T lymphocytes, and their job is to kill cancer cells. Well, just like cancer cells, in order to do what they do, they need food. In order to make more T cells, it's a warfare, and it's a number of soldiers. So you need, for every cancer cell you kill, you need a T cell. So you need to increase your T cell numbers. You also need to create, using a warfare analogy, arms. It's an arms race, and so in order to do that, T cells need nutrients to build it out. And the tumor intrinsically knows that, or evolves to learn that, and what the cancer will do is eat up all of the nutrients around so that the T cells don't have access to them. And in doing so, T cells are less effective. So that's one area that we've been really excited about, understanding where that nutrient competition lie. What are the things that the cancer cells are eating that, if left behind, that the immune system can now be more effective, and we found ways to block the tumor from having access to some of those nutrients. And what's remarkable is that when you do that, the tumor actually doesn't care, really indicating that the reason it was gobbling that stuff up wasn't for its own purposes, its own growth purposes, but to really keep those from the immune system. And when you block the ability of the tumor to do that, the immune system now can really ramp up and go after the tumor. So that's an area we're interested in. What are those cooperative sharing pathways? What are those competitive pathways? I'll tell you one other area. Got a lot of interesting areas in tumor metabolism. One of the other areas that's going to be close to your guys' heart. In some tumor types, including pancreatic tumors, there is what we call a microbiome. Right? So that microbiome consists of largely bacteria, but also fungus. And they are part of the tumor microenvironment. They're another cell type in the tumor that's making stuff, and that's influencing the way that the cancer cells and the other cells in the microenvironment are reacting. A postdoc in the group, Dominic Watt, is working on an aspect of the microbiome called the mycobiome, which is fungus that live in tumors. And he's found a fungus, so he's been working with our colleague, Dr. Neil Daly, and they've isolated pancreatic tumors from the clinic and duodenum from the clinic, and they have used some really clever microbiology techniques to streak out the microbes that live inside of those two things and then to grow those out. And so we have clinically relevant microbes, and he's been studying how those can be used to influence pancreatic cancer in our mouse models. Long story short, in his pancreatic cancer mouse models, if he uses antifungal agents, he can make the tumors grow slower. And if he repopulates with the microbes that he gets out of the clinic, he can make the tumors grow faster. So he's been working to understand what are the metabolites that the fungus, it's a yeast strain he's working on. What are the metabolites that the yeast strain is making that are influencing the tumor and how? And his current model is that they produce a variety of signaling-like metabolites that make the tumor. So what I didn't tell you is the immune system has sort of two faces. There's that which is the killer side, and that which is the protective side. So tumors tend to have a lot more of the protective side. It's the anti-inflammatory immune system, and that's what those metabolites from the fungus are doing. They are helping the anti-inflammatory immune system to be anti-inflammatory, and in doing so, that is just another one of the many mechanisms that keep the killer T cells away from the tumor. So we've been studying metabolism in cancer, but now we're kind of branching out to microbe metabolism. Super interesting area.
It's interesting. I've never heard of that.
Patrick's doing a lot of this too. He does it with he's been studying bacteria.
Oh, really?
I encourage you to read a cool paper from Hannibal on Ruterin.
Oh, I think I just met Hannibal recently, actually.
Yep.
Really cool work, yeah. Very cool.
You guys should know about this stuff, given that you are interested in engineering bacteria to go into tumors.
I'm really glad you brought up microbes too, because I can.
Good segue?
Great segue.
Okay, perfect.
So, as we discussed earlier, our project revolves around taking a magnetotactic bacteria, and using that to target ovarian cancer cells. Just broadly speaking, what challenges might you see with this approach? And if so, how might we best attack those?
Oh, man.
Just be as critical as you want.
Yeah. I mean, the challenges, I think the challenges that you guys are going to be really ready to and able to address are engineering the bacteria. And I think that's probably going to be the most straightforward aspect, and it's going to be that which you can really do in the research lab. So I think you're engineering the bacteria to express this heat shock inducible payload. I can't remember what you told me.
Cytolysin.
Yes.
The cytolysin. So you're in the process of doing that.
Yes.
Right. So the major challenges that you're going to face are putting that bacteria into a person, into an organism. Let's say when I put it into a person. Putting that bacteria into a person without creating some sort of like septic shock in an organism, right? How do you make it such that the microbe doesn't kill the host? How do you get it selectively to the tumor and not somewhere else? And you're not the first person or people to think about this. So there are, I don't know about this strain you're working with, but there are ways to make bacteria that have kill switches, so you can just feed them some sort of metabolite, and then it makes the bacteria die, or some other condition that leads to so you don't have to worry about the bacteria proliferating and damaging the host, or you can make bacteria that are somehow replication incompetent. That would be another way to make sure that they don't get in somewhere and overtake the host. That's going to be a challenge. My impression from people doing this is that it is a surmountable challenge. Getting the bacteria to the tumor is a challenge, but I think that one may be one of your more surmountable, at least insofar as maybe from a simplistic perspective. Tumors, I told you, are anti-inflammatory environments. They're immune-privileged environments by design. They want to keep the cells who kill stuff out of them, because if those cells get in, they're going to start killing cancer cells. So what are all the different ways that they can keep the killer immune system out? By being an immune-privileged space. So bacteria are going to land there by accident, and once they get there, it's going to be a safe harbor. In some instances, they will hone there. Tumors tend to be slightly more acidic than are the rest of the host for several reasons I can get into if you guys want to. So bacteria also tend to be attracted to that kind of area. I think we talked about hypoxic tumors earlier. Yeah. Or hypoxia, so some microbes prefer anoxic conditions to what the oxygen tension in the bloodstream will be. So that might be something that preferentially gets in there. So I think there's a couple of good reasons that you don't even need to use your magnets to steer them to the ovarian cancer. Once inside the tumor, how are you going to get it into the cancer cell? I think from 10,000 feet, your idea of using the folate transporter, I like it. I told you the devil's in the details. I don't even know what challenges you're going to come across. I suspect a bunch. Those may be surmountable too. People use folate receptor to bring in exogenous cargo, exactly for the reasons you've described with antibody drug conjugates. So I could see this working. I could also be, this is not my area, so I could be naive about possibilities, but it seems like, and then once you get inside the cell, how are you going to have the payload released / engaged? Right, so Arnav was telling me about a heat-based approach. Turn on the heat shock element-induced biosynthesis of this toxin. Okay, I think Devil's going to be in the detail there too. You may have wanted to started with something like a skin tumor, like a melanoma or a basal cell, so you don't have to worry about getting inside the body where the ovaries live. I think some of these skin cancers also express the folate receptor. So the folate receptor is expressed, it's higher expressed in a handful of cancers. I think there's a skin cancer that falls into that, so maybe something to continue thinking about. Let's see, the next step. So once the payload is delivered, created, is it actually going to kill the cancer cells? And cancer cells are really good about kicking out drugs. Mm-hmm. So what if all of the drug just gets kicked out? Mechanisms of resistance. But maybe you can kill them before they figure out how to be resistant. I don't know. I think it's very logical. It can be broken down into pieces too, so you don't have to study it linearly like that. You could be working on designing your bacteria to express the payload, and you could have someone else working on bacterial delivery to the tumor, and you could have someone else working on tumor cell uptake of the bacteria, and yada yada.
Yeah, I think those are really fair points. We're hoping to study some of them, for example, how well the bacteria is actually able to kill cancer cells. Down the line, once we have our final AMB1 host with all of our plasmids, we hope to do co-culturing with mammalian cells in my lab. So with that, we're going to be running Westerns to see the cytolysis SNA production, whatnot, and then also cell viability assays. Sure. Both control AMB1 without our plasmid, and then also with our plasmid to like you said, to see to what extent is the bacteria alone killing the cancer cells. Mm-hmm. But yeah, that's something we hope to do once we have our final AMB1 host fully constructed. Arnav
was telling me that was a 1-year program. You guys have extended it to 2. Is there a
Yes.
chance you're going to extend it to 3 and actually pursue some of these ideas with more, or does it die with you guys?
I'll be gone by then.
You don't care. That's your legacy. You gotta pass it on.
She'll still be here.
I'll be sticking around, but I do think we'll move on after this. We just our main reason to continue is because we know we knew what we did wrong in our last cycle. We knew our promoter was just off. We discovered that weeks before the competition, and there's not much you can do by then. But we really liked our project, and so we put a lot of thought into trying to come up with new project ideas. But ultimately we decided to stay with the Magnolice project.
Okay.
So I do think this is the last year. After this year, no.
I will. Okay. Well, fair enough. You may have made it more complicated. So you've decided to work with this magnetic bacteria because you guys thought it was clever because you could direct it with magnets to the host.
True.
Right? Okay, that may be true, and it is clever. I totally agree. I like that. But there are bacterial strains that do home. I don't know which one of those, N or M. I think it's hone. Will hone to tumors that are much more genetically manipulable and don't require exotic techniques.
Yeah.
So if I were starting this project de novo, I think I would have said, you start with that bacteria.
Mm-hmm. Yeah, I think part of our motivation to use AMB1 was because it's it hasn't been utilized that much before.
Okay.
Specifically for iGEM, I'm pretty sure Avi, our other science director, said that last year or maybe the year before in iGEM, the only other team who had worked with AMB1 just proved that they could kill it or something. It was a very surface level finding, and so I think that was our major motivation is because it's not widely utilized. So, yeah. But, yeah, I would agree that there are probably some better suited bacterial hosts. But, yeah.
I think if nothing else, it kind of goes to demonstrate what AMB1 can do. Because our goal, essentially, with using it is to... It makes it a little easier to have, a heat shock promoter within that bacteria.
Mm-hmm.
Obviously, having the magnetosomes and everything provides, another, I wouldn't say catalytic step, but another step closer towards reaching that critical temperature. And yeah, I think more or less it's just the concept of trying such a novel bacteria, just kind of showcasing what routes it can take.
Yeah,
Okay.
I think a big aspect of it was just the uniqueness factor. And then I don't know if Arnav explained this part or not, but the thing with the magnetosomes is the idea is that if you can put it in a machine that rapidly flips the magnetic field, we found that it can generate enough heat to activate that heat shock promoter. So that's where the heat shock activation is coming from, which is just another.
So you using this magnetotactic,
magnetosomes.
this bacteria for the purposes of making it hot, not for the purposes of directing it, or those.
I would say it's a
It's
combination
both.
of both
Okay.
factors. Yeah.
I totally appreciate the novelty and the science fiction factor. I love that. So, no shade from that perspective. I think it's cool. Is there intention of writing this up in some capacity? Taking all your hard work and putting it out into...
That has been brought up before. We have talked about it. We think it's kind of outside of the scope of our club. Because the goal of our club isn't to produce papers, it's, to compete in iGEM.
Yeah.
So that's been vaguely brought up before. But a lot of our members work in labs and, have publications and have agreed that our project can be published. But that's not something we've given much thought, I would say, as a team.
I would encourage you to consider it more. I think for any of you that want to go into science, I think it's a good exercise.
Mm-hmm.
Learning, thinking about how to present your findings in research manuscript format.
Mm-hmm.
You can, at the very least, submit it as a preprint on a preprint server like bioRxiv, and just put it out into the world, and the next group that's working on similar concepts can take advantage of what you learned,
Yeah.
and what you did right, what you did wrong, include all that in there with figures and so forth. And also, then you can list that as a line item on your CV that says, we worked on this project, It was, a tangible project because here's an outcome. You could go one step further and submit it to a journal too. That might be more than you're looking to do because that's going to require the peer review process, and then there's some memory that's going to be needed. So maybe you could pick up that in year 2. But is that something you want to do with your time?
I would really be excited to see where we could go in terms of like written work, because I think you make a fair point in regards to that. I think it'd be cool to see where we could go with like writing something up. Yeah, because we definitely learned a lot from this project, and there's definitely a lot other people could also learn.
Yeah.
It's like 2 years of like a lot of troubleshooting and stuff, so definitely very cool.
Well, if you need a hand in that regard, let me know.
We really appreciate it.
Glad to offer some feedback. You got Yatrick and Liang, and I mean anyone in the Shaw Lab can show you what it's like to write a paper. So
it's always good to have like insight from different, places though, for sure. So we really appreciate your insight.
Yeah, of course, my pleasure.
And it was great having you.
Yeah, thank you so much.
If you could give like one word, a single word of advice to any future scientist, what would it be?
One word is hard. I think persistence.
Okay.
Yeah.
You're going to experience a lot of failure in science.
Yeah, especially in research, right?
In research, yeah.
Experience,
That's what makes it fun. If you knew what to do going in, it wouldn't be interesting. 9 out of 10 things you try aren't going to work, and not because it wasn't a good idea, not because you weren't clever, and not because you didn't try hard, just because it didn't work. So you need to be persistent.
That sounds good.
All right.
Thank you so much.
My pleasure. Good chatting with you guys. Nice to meet you
So nice
both.
to talk to you.
Keep in touch, huh?
For sure.
Yeah.